right, welcome back to another exciting episode in the woodshed here. We are going to be discussing mutations here, and in just a second I'll show you those details. Actually, let me switch camera angles. This is kind of inefficient, if you will. Here's our section again. I'll go over that in just a second. It's going to be about mutations. And I wanted to start with kind of a worship thought that I think is really relevant. So I just got real excited about this as I sat here and read this for about 20 minutes before I came back to you guys here to start this video. But I was just going to read one verse about how, you've heard the verse in Romans about how all things work towards the good, right? How God can, this idea that God can take what's bad and work it towards something that's good. I think that's an awesome idea and concept, that God takes our mess and cleans it up. Like, man, and he's so humble about it too. He's kind of like the people that, I don't know if any of you have ever stayed late at RAA and seen the people that go around literally every classroom and take out the trash and pick up stuff, food boxes that are on the lunch tables, which they shouldn't have to do, by the way. But they do all the dirty work. They clean up our classrooms, and they do it in the evening and no one's there. And when I stay late, I see them, and I just think, man, like, what a servant, someone going back and doing all that cleanup so that we can all come together in the next day and learn. And they don't get any, well, they get paid for it. It's not like they don't get credit, but they don't get any acknowledgement, really. People that do that kind of work often don't get acknowledgement, and God does that with us, which is so cool. We've messed up our lives, and he's gone back and cleaned it up. And so what a time to reflect on that with Easter, and the sacrifice that he's made. But there's a really cool verse here in Romans that I wanted to read about that, how God takes what's good, takes all things, and they work towards his good. But I backed up, and there's some verses before it that I want to read, and I actually want to read it from the Message translation. This is such a powerful paraphrase. Remember, this is a paraphrase, not a translation, direct translation. It says, "All around us we observe a pregnant creation. The difficult times of pain throughout the world are simply birth pangs. But it's not only around us, it's within us. The Spirit of God is arousing us within. We're also feeling the birth pangs. These sterile and barren bodies of ours are yearning for full deliverance. That is why waiting does not diminish us." Right? So back up. The sterile and barren bodies of ours are yearning for full deliverance. Right? We're expecting that. And that is why waiting does not diminish us any more than waiting diminishes a pregnant mother. We are enlarged in the waiting. We, of course, don't see what is enlarging us, but the longer we wait, the larger we become, and the more joyful our expectancy. Meanwhile, what just happened there? Meanwhile, the moment we get tired in the waiting, God's Spirit is right alongside us, helping us along. If we don't know how or what to pray, it doesn't matter. He does our praying in and for us, making prayer out of our wordless sighs. That's so related to what we're going to talk about with mutations. He does our praying in and for us, making prayer out of our wordless sighs, our aching groans. He knows us far better than we know ourselves, knows our pregnant condition, and keeps us present before God. That's why we can be so sure that every detail in our lives of love for God is worked into something good. All right, so sometimes we don't have all the answers, but when we have faith in God and we're walking in his light, and we're making prayers, sometimes we don't even know what to say. We don't even know what to say. It's just aches and groans within us. It's okay. It's okay, because what that verse says is that through his Spirit, he makes intercession for us, and he prays those intercessions to God. So we don't have to have those words. Don't feel like your prayers have to be perfect written essays, right? God knows how to interpret. And that's what today's lesson is about, all right, is how the cell interprets, or what happens when the message is messed up, right? Because the message will get messed up. And so here we are. We're in 13.3. Now I'm actually going to switch camera angles here just so you can get a better image here. Hopefully this works for me. Yes. All right. So let me back up here just slightly. We're in 13.3, which is about mutations. And you have two different types, basically. I've kind of just summarized the information. I'm going to have to write in there in just a second. Maybe I'll do that at the end. 13.3 is about mutations, and you have two basic types, gene mutations and chromosomal mutations. And if you remember, genes are sections of DNA that are found on the chromosomes that code for certain traits. Genes are just small segments of the chromosome. Chromosome has many genes. And so, of course, you're going to have two different types of mutations. Gene mutations, which are focused on just the genes, or whole chromosomal mutations. Right? That doesn't sound like a good story, does it, to have a whole chromosomal mutation? But there's even a little bit more narrowing we can do there. With gene mutations, you have two different types. You have point mutations, and you have whole frame shift mutations. And so there's one type of point mutation, and there's two types of frame shift mutations. But again, that's what I'll go back and write that in, maybe towards the end when we do a summary, just to make sure we've kind of covered our learning objective. I didn't really write this in a sentence, articulating what you're going to do or what you're going to learn. I just kind of did a little summary here. Sorry if you're more interested in the essential question or learning objective. But the second one, the chromosomal mutations, we basically have four types: deletion, duplication, inversion. How do you like that for inversion? And translocation. So these words kind of speak for themselves. If you know what those words, delete, duplicate, invert, like I just did with the screen, translocate, if you know what those mean, you should do pretty good, because these words are kind of self-explanatory. All right, so back to this camera angle. Now what I'm going to do is I'm going to share my PowerPoint presentation with you through my desktop computer. So let me do this now. We are on camera 4. Yep, there's the Tunnel of Doom. That's my software I'm using. So let me minimize that screen, and let me minimize this, and let me open up my okay, great. So we do have a couple key questions. What are mutations, and how do they affect genes? Okay, so what are mutations? Well, mutations are going to be changes in genetic information. Well, what is genetic information? Well, you already know now that it's DNA. And in this chapter, you're learning how DNA can be translated. It's actually not directly translated, is it? It's first transcribed into RNA, which then that RNA is translated. But nonetheless, the original information was found in the DNA, right? And so if you have any changes in that DNA, you're going to have a mutation. In fact, that happens a lot, okay? And sometimes it's seen, sometimes it's not seen. If it's seen, that means the mutation is still there. But oftentimes, believe it or not, I'll just go ahead and say this now, your cells, when they copy the DNA, mutations are happening like all the time. I have a TED Ed video that I'll show you that really is really nice, short, easy, simple, and will help reinforce this material. And it has a pretty cool fact about actually how much your DNA is mutated. But what's really cool is you have enzymes, and you have processes in the cell that will help correct those mutations as they happen. But if they don't get corrected, this is what you get. You get look at that flower, that what is that, a daisy or sunflower? It's not a sunflower because those are usually yellow petals, right? With the black in the middle. But these are I think daisies, or some type of daisy. And look how it's supposed to be round, but look how it's all elongated like that. Look at the tiger, sorry, not the tiger, the lion on the right. The lion that doesn't really have the typical color you would see. That's because it does not have pigment really in its hair, skin, or eyes. So it would be like an albino. And so those are going to be traits that are visible, which, if you remember from chapter 11, was phenotypes. Those are phenotypes that are resulting from a mutated genotype. How about that for vocabulary? Let's look on to the next page here and see what type of mutations you have. So again, we said that there's going to be gene mutations. Let's make sure you can see all this here. Yeah. Okay, so you have gene mutations, and you're going to have two types: point mutations and frame shift mutations. Well, first of all, remember we're dealing with just a gene. We're not dealing with the whole chromosome. We're dealing with a section of a chromosome, a section of DNA that will code for just one trait or one protein. And so that's called a gene, that section of DNA. Well, let's say that DNA gets transcribed into RNA, and so that we have these RNA sequences here, or this RNA sequence, AUG, CGU, ACC, and UUU. And so those are going to code for the following amino acids. AUG codes for methionine, which is our start. AUG is the start codon, and methionine is going to be your first amino acid. CGU is your next codon that codes for arginine, and ACC is your next codon that codes for the amino acid threonine, and then UUU is your next codon that codes for the amino acid phenylalanine. Now, typically this is going to keep going on and on and on until you finally reach a stop codon, and this is actually going to be anywhere from 20, 25, 30, 40 amino acids long, right? Much longer than 4. This is just kind of a simplified example here. All right, so we have 4 amino acids that we're working with in this example. This would technically qualify it as a polypeptide because we have more than one amino acid, and so it already meets that qualification of being a polypeptide. Remember, poly means many, so we have many peptide. These are actually peptide bonds here that are connecting the amino acids. The amino acid, if you remember, is the monomer, and the polypeptide would be the polymer. This would be the polymer. And then once it gets done being folded and twisted, it'll be turned into a protein. And so sorry for that little tangent. I just wanted to reorient you if you're unfamiliar with what we're looking at here. We're not just looking at colored circles. Okay. So we have the intended sequence of amino acids here. Well, what happens when you go through the following mutations? The first one is called substitution, and it's a type of point mutation. It's the only one, right? And so that's one of the words I'm going to end up writing on my chalkboard again at the end when we wrap all this up again. Under point mutations, you have substitution, where basically we used to have a C right here, but if you take that C out and let's say the enzyme just made a mistake and actually put thymine there instead, what would you end up with? Well, you would end up with this sequence of nucleotides in your RNA, and you would end up, which everything's the same except for where this substitution is, right? And you would end up with this codon, CAU, because you'd have A here because you now have T. We used to have C, so we used to have G there. So this used to be CGU. If you don't believe me, you can just look up here, CGU. But now it's CAU. And so what does CAU code for? Well, if you look at that codon wheel, CAU, just to recap for you guys, how do you use that wheel? You go C, if you start from the inside and work your way out, CAU, that codes for histidine. Histidine. Well, that's kind of a problem because the amino acid that's supposed to be there is arginine. So we have a problem. I mean, yeah, we still have threonine here and phenylalanine here, but we have a different sequence of amino acids, which eventually means this different protein, okay? And so that can be a problem. Now, that can be fixed, though. Again, there's enzymes and things that happen in the cell that will help fix that if that happens and put the C right back where it was so that this codon can then be CGU instead. But that's one type of problem you could get, okay? And again, remember, this makes a totally different protein if you switch this amino acid and becomes histidine instead of arginine. The red circles are going to represent new amino acids that are going to change from the normal situation up here. Okay, so the next two types, insertion and deletion, are going to be frame shift mutations. Frame shift. And those are kind of deadly because it's literally shifting the entire reading frame. And I'll show you an example of what that looks like using typical words, the language that we read, right? This is the language for DNA and for your cell. This is the language for your cell to make proteins. And well, if the ribosome specifically is reading this sequence of this sequence of nucleotides found on this mRNA strand, well, it's going to get totally different proteins if you just all of a sudden insert a nucleotide in the DNA. So you see up here in the DNA we had TAC, GCA. We still have TAC, GCA, but now we're going to insert an adenine in between the cytosine and guanine. We're not taking anything out. We're not substituting anything. We're adding a nucleotide. We're just adding a nucleotide. We're squeezing it in between. It's like somebody cutting in line. And so in this case, when the mRNA strand is made, then you're going to obviously follow base pairing rules, and where you see adenine, you're going to put a uracil. But now look at your codons. Now you have UCG. Everything got shifted. Everything got shifted. So not only is this codon messed up, but so is this one, because the last letter here got bumped over to the next one, and then the last letter that was there got bumped over. Everything got bumped over. So now our whole sequence is off. The only amino acid that's original is the methionine to start with. So insertion, huge deal. That's a huge problem because it ends up shifting the whole reading frame. All right. Another one would be deletion. Let's say, again, we start with TAC, GCA. Down here we have TAC, GCA, but we take that G out. Then all of these nucleotides get shifted over to the left. Well, if that happens, then again, now your codon sequences are going to be different. You're going to have GUA, CCU, UA, and now you're just missing something here. Remember, you need all three nucleotides here to make a codon. So you can see everything past this mutation is going to be off. Now you're still going to code for amino acids. The cell doesn't know the difference. It just reads, right? It just reads, but it's not going to end up making any sense for the cell. And so you're going to have problems. And so insertions and deletions are going to cause entire frame shift mutations. All right, the framework that you're reading from is going to be shifted, and it's going to be a huge problem. And so here's an example. I just typed these out just a minute ago. Let's see if we can figure out where is the mutation and what type is it. Let's look at the top one here. Where is the mutation and what type of it is it? Well, that sentence makes sense, and so there's obviously no mutations there. I didn't intend to have any mutations there, so that one's kind of the baseline that we're going off of. Let's say that we want all these sentences, right, to look like this one up here. Well, let's look at the next one. Where is the mutation and what type is it? Where in the mutation? You see how I read that? It's like I didn't even know. It's like I just passed over that and just barely, because it looks kind of similar, doesn't it? What happened here? This is a substitution. I just substituted the S for the N. And so it kind of makes sense, right? And oftentimes mutations like that aren't necessarily always going to have a huge effect on the cell as long as it can, as long as the rest of it kind of makes sense. But however, this doesn't quite make sense though, does it? Because in is a different word than is. Even though they kind of sound similar, this sentence does not make sense when you say where, or when you ask where in the mutation and what type is it. That doesn't technically, following the rules of English, make sense, right? Now you could say, where in this sentence is the mutation? You could say something like that, but the way we have this sentence, it doesn't make sense. And so technically it is wrong, right? Even though it kind of looks like it. We've substituted the S for an N, okay? How about this one? Where is the, it's all gone wrong now. Where is the mutatio indua hot EPI is it? Right? That doesn't even make sense. I can't tell what's happening here. But guess what? If you were allow me to, let's see, maybe put mutation. If you were to allow me to put an in here, if you were to allow me to put an in right here and shift all of these characters one over, then we'd be right back where we started from, and the rest of this would make sense. You see how it kind of just all goes downhill from right here? It's because we're missing an in. So what happened here? That would be, first of all, it would be a deletion. It would be a deletion. You deleted the in. You took out the in, but you didn't replace it with anything. So you just deleted a nucleotide from the DNA, and then after you translated it, you can just see in the translation process, everything gets messed up because it's a frame shift. All of this framework for reading got shifted over one, okay? And so, how we have words, codons are kind of like words. Each word is a word, and you can't really just, It's not like we just have a giant long run-on sentence. And so if you take a letter out, if you move all the letters over in every word over to the next word, then you mess up the word before it. And so again, this is a frame shift mutation. And so one mutation here causes the rest of the words to not be legible or translatable anymore to anything that makes sense. And so that's exactly what happens in deletion. It's exactly what happens in a deletion mutation. All of these amino acids, they might be there, but they won't make any sense. You're not going to end up making the protein you want to make. It's just going to be some random sequence of amino acids that'll make some random protein. Now, we'll talk in just a moment of how that could be a good thing, because it may be a random protein, but it might be a beneficial one. So how about this next one? Where is the what's happening here? What is that S? Where'd that come from? That's an insertion. That's an insertion. And it looks like we're missing the N here, but we're not. It got moved over here. Well, then that made this D get moved over from and to over here. That made the T that was in the word what get moved over. It made the E that was in the word type get moved over, and so on. So you can see just inserting a nucleotide or inserting a character where it shouldn't be moves everything over. So these two types that we've just looked at are going to be frame shift mutations. Those are bad. How about this one? Where is the mutation and what type is it? This one's interesting. I did this one on purpose. We didn't really have anything too crazy going on here. This is just simply a substitution. We substituted the letter A for the letter E. Well, we still kind of have something that makes sense, right? And believe it or not, that can actually happen even in the cell, because if you look at your codon wheel, you can see that there are more than one codon for the same amino acid sometimes. Like, for example, here's a great one, serine. You can have UCU, UCC, UCA, and UCG to code for serine. So simply replacing the last letter here, like the or the, it doesn't matter. You're still going to get serine. The cell is still going to make serine. It's still going to drop off the amino acid serine when it's making that polypeptide. So sometimes the substitutions aren't always bad, because they won't really change anything, right? It's just a different way to spell the word the, right? Like some people text now with that. They'll just text the word the like that, and that's okay. Like, we all understand that, and we have almost accepted that now, right? Okay. The next one, the big separation here with mutations is chromosomal. So with chromosomal mutations, you're not just affecting the gene, you're affecting the entire chromosome. So let's assume this whole section here of genes is a chromosome. And let's actually say that A, B, C, D, E, and F are independent genes. So A is an entire gene, B is an entire gene, or let's just say an entire portion of the chromosome, okay? And so with chromosomal mutations, they involve changing either the number or structure of chromosomes. So you can literally actually change the number of chromosomes, and that's kind of a terrible mutation. Or you can change the structure of a chromosome, which could be pretty bad too. But it's almost kind of like reading the end of a book before the beginning of a book. You still have all the book, and you still can read the chapters. It's just kind of out of order, and so that could cause problems, but could not, So these mutations can change the location of genes, right? So you're just like moving the genes around, or they can change the number of copies of some genes. And so we have 4 types. So let's look at all 4 of these different types of chromosomal mutations, and you should be able to answer test questions related to these different types. So deletion. With deletion, it's just like taking a whole gene out. So you see how you have gene here, B? Well, if you just take that out, here's your new chromosome. Now it's just A, C, D, E, F. Well, where is B? It's gone. So guess what? In an organism, there's not going to be that genotype anymore for B. So again, you won't have that phenotype. Whatever that B gene is, whatever the protein that is made for that from that gene, from that information in the gene, is not going to get made. So that protein will not get made, period. All right. Now you also have this one. This one's interesting. Duplication. You literally just get, let's say that gene got copied twice. Let's say for whatever reason during DNA replication, which is, by the way, when this usually happens, when DNA is opened up and then copied. usually it gets photocopied wrong. Like, let's say you're in a rush and I'm at the copy machine and I'm in a rush and I'm photocopying page after page. Sometimes I literally forget to copy a page, and there'll be like a 10-page test, and one of the pages I just forgot to copy or just missed it somehow because I got stuck together or whatever. Or let's say I accidentally copy a page twice. Is that a bad thing? Doesn't seem like it would be, right? Because you just basically are going to have an extra page. So you can just kind of ignore one page. And that's kind of what the cell could do as well, because at least the information's there, right? Inversion. This is an interesting one too. Inversion means to flip or to reverse. And so instead of going A, B, C, we invert B and C so that it's A, C, B. But it's okay because look, the gene for A is still there, the gene for C is still there, and the gene for B is still there. So not that big of a deal, really. Probably. I can't say always. You can't, because sometimes genes go together. B and C, or B and D may have gone together. Or no, say originally C and D may have gone together as a group, and they needed to go together as a group. But if you invert it like this, now C is no longer right next to D. C is over here, and D is way over here, and so it causes problems. So it's almost just better, let's not have mutations in the genes. That's kind of the key, that we don't really want mutations. Well, we'll get into that conversation in just a second. But finally, the last type of chromosomal mutation is translocation. So it's taking something from one place and moving it to another. So literally, let's say you had G and H on some other chromosome way over here, and you took that out of that chromosome, brought it over, and translocated it to sandwich in between these genes. So maybe these genes were for, I don't know, height. Well, now you're taking them from the chromosome that they were on and you're putting them on a whole new chromosome. Well, may not be a big deal either, because at least the information's there. But you remember, you have 23 chromosomes, at least in the human cell. And what happens if you take them off and then you go through meiosis or mitosis and they don't get moved into one of the cells? That could be a problem. So here's your 4 different types of chromosomal mutations. You have deletion, duplication, inversion, like a switching, and translocation. So memorize those. Also, this is a quick lab. I'll create an assignment on Google Classroom so you know to do that. There's a little exercise kind of similar to the one we just did with the words that I did, but this would be your own little just quick lab. It's not really a lab, but it's 3 questions that'll allow you to kind of get hands-on with this material. Finally, before we wrap it up, we just have 2 more pages, and then you guys can do this assessment also for a homework assignment, just to answer questions. So what causes mutations? The word we use is a mutagen. A mutagen is just simply a Latin word that means origin of change. It's a place of change, an origin of change. It's where change originates. And stress, it's usually can come from many different places. So stressful environmental conditions can actually cause some bacteria to increase mutation rates. And this can actually be helpful to the organism, since mutations may sometimes give such bacteria new traits, such as the ability to consume a new food source or to resist a poison in the environment. And so we're answering the question essentially here: how do mutations affect genes? Well, again, if you remember what genes do, they code for specific polypeptides, which are eventually going to be proteins. And changing these, mutating these sequences, is going to mutate the sequence of amino acids, which is going to make a different protein. And so you're going to get different traits. Well, you're going to get different proteins, but that means you could get totally different traits. But it's not always just going to be a different protein that'll still code for a variation of a trait. It may code for a protein that just doesn't make any sense and have anything to do with the trait. And so basically that trait just doesn't get expressed at all. And so so they're going to be, in my opinion, they're errors in the genetic process. When DNA replicates, usually, so when DNA is replicated, that's when there's usually going to be one of those errors. and the cellular machinery that replicates DNA inserts an incorrect base literally roughly once in every 10 million bases. But small changes in genes they can gradually accumulate over time. And so again, these things that cause mutations, where can they come from? Where do they come from? Like we said, it can happen in the copying of DNA, but where else can it happen? Well, there's two different places here: chemical and physical origins. So you have chemical mutagens, which those are going to be origins from chemicals like pesticides or even natural plant alkaloids, or tobacco smoke, which is natural as well, right? But they weren't intended to be smoked, and so it's not a natural way for you to get plants. And again, even just because it's a plant doesn't mean you can eat it. You can't eat every plant. So a lot of chemicals or environmental pollutants, which are chemicals, can be a source of mutagens, chemical mutagens. When you have physical mutagens, where they're not necessarily chemicals, but physical processes like forces from electromagnetic radiation, right? Now, cellular radio waves that power your cell phones or used for communication, they're not going to necessarily, well, it depends on who you ask, but definitely it's electromagnetic radiation that's powerful, like X-rays or ultraviolet light from the sun. Those will cause damage to your DNA, right? They're forces that will mess up your DNA. They will destroy it. And if these agents interact with the DNA, they can produce mutations at super high rates. And cells can sometimes repair that damage, but sometimes they cannot. And if your cells can't fix the damage, then guess what? You're going to have changes that are permanent. And what's going to happen to that DNA molecule? It's going to get copied, right? And then you're going to end up with a bad copy. And then that copy is going to get copied. And so there you go. It just keeps continuing. So imagine like I made a test, right? Imagine I made a test, and then somebody let's say even me, myself, I accidentally had a marks a lot and just accidentally gone across like all of page 2 and just messed up all the questions because it was like dark marks a lot and just all across the questions. Well, now when I go photocopy that test into the machine, I put 40 copies back to backs, please staple or whatever, and hit enter. I'm about to get 40 copies that are all messed up. And if I wanted to make more copies from those photocopies, that mark's always going to be there, right? And so we have to be careful with permanent damage from mutagens like that. Like UV rays can do that, X-rays, and so and those can end up causing cancer or other harmful effects. What about helpful mutations? Is that a possibility? Sure. Theoretically, the effects of mutations on genes, they're going to vary widely, and some have little or no effect. Some can produce beneficial variations, and some negatively disrupt gene function. I'm going to say the last one, this is just my opinion, is that most of them negatively affect the gene function. We were designed a certain way, and I think anything that is a mutation to that gene, it could possibly have a beneficial effect. I'm not saying it can't, but I think that's just a coincidence, or you just got lucky, because generally any change to our original design, as far as mutations, and I believe in variation, genetic variation I don't think the genetic variation only comes from mutations. I don't believe that. Or at least if there is, God found a way for that to happen naturally through certain things. But going out in the sun and baking your skin and then getting you're going to get skin cancer, all right? Or ingesting chemicals. That one's a little harder for me to argue with, because like I said up in this example, it said that certain insects have gained resistance to pesticides. Actually, it's right here. The beneficial effects it's here. It says that some mutations have helped many insects resist chemical pesticides, and some have enabled microorganisms to adapt to new chemicals in the environment. And so, for example, even mosquitoes, the mosquito genome has made many African mosquitoes resistant to the chemical pesticides once used to control them. And so, yeah, that's one of the tougher arguments to make. That's a pro-evolution argument, that mutations are what cause organisms, period, to evolve. But I don't personally think that mutations have caused us to go from one-celled amoebas. The chances of even us becoming a one-celled organism just randomly and arbitrarily are pretty high. But then that the mutations would allow us to be who we are now, I'm not buying into that. I'm sorry, but that's the thought. It's just so you know. So when you're in those biological circles, you know what you're talking about, and you know what people are going to be coming at you with. All right, that's their reasoning. That's the general exception for why species evolve, is through mutations in DNA. All right. And then once those mutations happen, the survival of the fittest happens. The organisms that are most fit for survival will reproduce, and those mutations basically just get passed on from offspring to offspring, right? And so here's another harmful effect, an example of a harmful effect on, let's say, the red blood cell. You make one small chain in one of the polypeptides. The red blood cell is actually made up of 4 polypeptides. Well, if one of those polypeptides has some mutations, then you end up with sickle cell anemia. If you've ever heard of sickle cell anemia, it means when the red blood cell has this shape, instead of being a nice round shape like these, they have these funny shapes like this, like a crescent moon or whatever, or flat. And basically, that means that protein is not going to be able to, the hemoglobin's not going to be able to do its job right. And so you're not going to be able to carry oxygen. That's what red blood cells do, is they carry oxygen throughout your blood. Excuse me. And so sickle cell anemia can obviously cause major diseases. So you can get severe pain because oxygen is needed for your mitochondria, remember? And so it's just going to cause a whole lot of issues when you don't have red blood cells adequately carrying oxygen to your cells and your mitochondria, because if you're not making ATP, it's kind of like everything else shuts down, right? So it's kind of like this snowball effect you see right now in our economy. If ever we shut down fuel sources and transportation, everything else kind of shuts down. It's kind of a chain reaction. And so sickle cell anemia is bad, and it's basically a point mutation in one of the polypeptides found in hemoglobin, which is the blood's principal oxygen-carrying protein. All right, now here's some, again, beneficial effects. We've already kind of read that, so I don't think the only one they added here was you could coincidentally have mutations that would increase your bone strength and density, making fractures less likely. Or you could have one that increases the resistance to HIV virus that causes AIDS. Now, just so you know, that's not that great, because you really have literally like a 100% chance, basically, 99.99, let's say, percent chance of not getting HIV if you don't use needles and you don't have unprotected sex with people that have HIV. Then you're not getting HIV. Like, it's pretty easy not to get HIV, right? You have to do some pretty sketchy stuff in order to get HIV. If you're not taking doing what I just said, you're not going to catch HIV. You don't get it by coughing, so you're exposing your blood. So make sure you're not having any blood transmission. But again, too, you don't just have a cut and you touch someone else's cut and you got HIV. That's the virus doesn't survive that long outside of outside of the body like that. So, but it can. That's possible. But I'm just saying, unless you're using needles and to do drugs, or you're sharing needles for other reasons or whatever with someone that has HIV, or you're having unprotected sex with someone that has HIV, you ain't getting it. So, just so you know. Anyways, plant and animal breeders, they can make use of some beneficial effects. This is the last thing I wanted to bring up before I end this video. There's something called polyploidy. Do you remember when we talked about meiosis? How we said that sex cells or gametes are haploid, and they come together with another haploid gamete to make a diploid organism. All your cells are diploid except for your sex cells. Sperm and egg are the only cells in your body that only have, I don't want to say half the number of chromosomes. They don't have a paternal and a maternal version. They still have all 23 chromosomes. You have to have all 23 chromosomes or you don't have a full book. Remember, the book is the analogy I like to use. But for most of your cells, you have a mom version and a dad version of those books, all 23 chapters. All 23 chapters written by mom and 23 chapters written by dad. Well, you can actually have 23 chapters but have a mom, a dad, and another version, and that's okay. You can do that. And so here's some pictures I found that might illustrate this, because your book uses this example of citrus fruit being that way. And so I thought it'd be cool to just do a further exploration, since we're in Redlands and we're in the citrus grove community, right? We do a lot of citrus growing here in Southern California. And you can see that technically these are the only 4 original citrus fruits. The pomelo, you've probably seen those in the grocery store, the citron, the pepita, and the mandarin. Here's the taxonomy names, genus and the species. They're all citrus genus. The species are different. You have Citrus maxima, Citrus medica, Citrus macrantha, and Citrus reticulata. And when you take, so there's like 9 chapters. You know, we have 23 chromosomes. These have 9. You have 9. You have 9 from mom, 9 from dad, 9 from mom, 9 from dad, 9 from mom, 9 from dad. Well, if you cross a citron with a pepita, you can get a Mexican lime. If you cross a citron with a sour orange, you'll get a lemon. But to get the sour orange, you actually first had to cross a pomelo with a mandarin. So it's thought that these are the original citruses, and everything else that we normally enjoy, like oranges, lemons, and limes, are all fruits that come from a tree that, if you look at the cells, are actually going to have more than one copy of the chromosomes. You're going to have copies from these, more than one organism here. Here's another, I thought, pretty cool flow chart, because I couldn't find the orange. I only saw the sour orange on this one down here. It said sour orange. I'm like, well, what about all the sweet oranges? I have a sweet orange tree in my backyard. It's the navel orange, by the way. And so I was like, what about those? Well, if you take again the Citrus maxima and the Citrus medica, you get the sour orange. But then if you do some interspecific hybridization, so just more interspecific hybridization among the oranges, you can get a sweet orange, right? So there's a lot of orange crossbreeding. And then this is one of my favorite, Meyer lemon. Those are real popular in Southern California. If you can get your hand on a Meyer lemon, it's one of the best lemons versus your standard lemon here. Bergamot also is an interesting one. They actually use the oils from that in a lot of colognes and stuff like that. But cool. I think that's going to be it for this lesson. So just make sure everything went well. My recording went well. Great stuff. I had fun with this lesson. Hopefully you guys understand mutations and the different types. Just to be clear, I told you I'd wrap this up with finishing my chalk board here. So for the point mutations, we had substitution. All right. Now for the frame shift mutations, the ones that are kind of a bigger deal because they shift the whole reading frame, you have insertion, and you have deletion. You insert a nucleotide or take out a nucleotide, and you're in big trouble because it's going to shift the whole reading frame. But if you just simply do a substitution, might not be so bad. But again, all of this depends. This is just setting the groundwork for just for you to understand some of the terms and how this works. But really, you could read a whole book. You could take a whole class on mutations, and it's all going to be specific to what protein you're even looking at, all right, or what gene or what organism you're looking at. And so this stuff is very complex, but at the base level, hopefully the vocabulary makes sense to you and the idea of what a mutation is makes sense to you. And we got one more lesson. We're almost there. Thanks for watching.